Hello, everybody. I think we can get started. Welcome to this parallel session, Room B. I remind that every speaker will have about 12 minutes for their talk, and the general discussion will be held at the end of the 3 presentations. We need to make a change in the order of the presentations, and we will have as a second paper the paper by Alexander Erler, and as the last paper that by Casartelli and colleagues. We have 3 papers today, all with long and catching and evocative titles. And the first paper is by a large research group whose members are Palumbo, Mariotti, Vellucci, Antonelli, Anderson, Harenski, Kiehl, and Pellegrini. And the title is "The Sins of the Fathers Do Not Fall on the Children. Selected dopamine and serotonin alleles increase the risk of psychopathy as a consequence of paternal maltreatment." I'm not sure who'll be presenting. I guess Dr. Palumbo.
Yeah.
Okay. Yeah, and the floor is yours, please.
Thank you. Psychopathy is a complex developmental disorder characterized by a combination of interpersonal affective problems, such as glibness, shallow affect, lack of empathy, guilt or remorse, and antisocial. Is there a way I can hide the column on the right side?
We don't see it.
You can move it with your mouse.
I see. Perfect. Yeah. All right. And social deviant behaviors, such as irresponsibility, impulsivity, aggression, and antisocial behavior. Psychopathy strongly predicts antisocial behavior. In the US, up to 25% of male prisoners meet the criteria for psychopathy, compared to 1% in the general population. Psychopathic offenders commit the majority of violent crimes, are resistant to treatment, and more likely to reoffend than other inmates, thus representing a major challenge for the criminal justice system. Over the past 20 years, neuroscientific research has begun to unveil the neurobiological and neuroanatomical features underlying psychopathy. A recent meta-analysis showed that psychopathy is associated with altered functionality of different brain areas, such as the amygdala and region of the medial default mode network and silence network, and to structural changes as observed in both criminals and general population. Genetic studies on twins and adoptees show that genetics explains up to 69% of psychopathy score variants, while the rest has been attributed to environmental factors. Psychopathy risk seems to be particularly higher when a genetic predisposition interacts with a negative developmental environment. To date, only a few studies have investigated the genes implicated in psychopathy, showing the association of 2 gene variants of serotonin pathway, 1 gene variant of the dopamine of the oxytocin pathway, and 4 gene variants of the dopamine pathway. These data are supported by further association between gene variants of the same pathways and juvenile callous and emotional traits, which are characterized by shallow affect and lack of empathy and guilt, predictive of some of the emotional deficit observed in adult psychopathy. Concerning the environment, it has been shown that early traumas, including prenatal, perinatal, and postnatal stress, are associated with callous and emotional traits and antisocial behavior, and may increase the risk of psychopathy as well. For instance, postnatal traumas like bad parenting, such as over-control, neglect, and abuse are associated with callous and emotional traits and seem to have long-term effects by increasing also psychopathy susceptibility. As the family environment represents the most important postnatal environment, the quality of parenting plays a key role in the development of behavior. To deepen our understanding of the complex gene and environment interplay in the risk for developing psychopathy, we investigated a number of candidate gene variants of the serotonin and dopamine pathways in interaction with parenting style in 232 USA adult white male inmates. Criminals come from an ongoing collaborative project with Professor Ken Kiehl at the University of New Mexico. He is known for having collected a large database of structural and functional brain data in addition to psychometric data of psychopathic criminals. To this aim, he put an MRI machine in a truck, allowing him to collect this data directly in jails, overcoming the problem of taking criminals out of prison to the hospital, where MRI machines are usually located. Thanks to this collaboration, we had the opportunity of working on a large data set of criminals screened for psychopathy by the Psychopathy Checklist Revised, and for parenting style by the MOPS, Measure of Parental Style, that measures maternal and paternal over control, abuse, neglect during the first 16 years of life. A sample of saliva was available for each subject. DNA was extracted from saliva and genotyped for 5 gene variants of the serotonin pathway and 6 gene variants of dopamine pathway. A stepwise linear regression analysis showed that paternal MOPS scores produced a significant model that explained 4% of the variance of psychopathy, while maternal MOPS scores did not contribute to the variance. After including genetic data into the analysis, we observed that psychopathy variance explained by paternal MOPS scores increased to 7% in interaction with HTR1B TT genotype, to 10% with ANKK1 T allele, and to 14% with THGG genotype. Furthermore, in carriers of both ANKK1 T allele and THGG genotype, paternal MOPS scores explained 35% of psychopathy variance. Finally, this data may suggest that there is a synergistic action between these two dopaminergic alleles in making the carriers more sensitive to paternal maltreatment. Finally, in carriers of all the 3 genotypes, that is ANKK1 T allele, THGG genotype, and 5-HTR1B TT genotype, paternal MOPS scores predicted 43% of psychopathy variance, suggesting a further additive contribution by the serotonergic genotype. In summary, we observed that paternal maltreatment increases the risk of psychopathy, and that the 5-HTR1B, NKK1, and TH gene variants interact with paternal maltreatment, increasing further the risk of psychopathy. The correlation between paternal maltreatment and psychopathy becomes stronger as the number of risk allele increases, as well as the variance increases up to 10-folds as compared to the one observed without including genotypes in the analysis. Overall, our results show that while paternal maltreatment per se has a minimal influence on psychopathy, when combined with the genetic profile, its influence becomes 10-folds higher. In line with our findings, previous studies show that childhood adversities induce epigenetic changes in HTR1B gene. As presynaptic 5-HTR1B exerts an inhibitor control on serotonin release, an increased availability of serotonin mediated by the TT genotype may affect amygdala responsivity to aversive cues and may play a role in the pathogenesis of the emotional dysfunctions distinctive of psychopathy. Also, the observed interaction between ANKK1T allele and paternal maltreatment is consistent with previous findings, showing that T allele carriers are more sensitive to rearing environment than CC genotype carriers. The T allele is associated with reduced expression of dopamine D2 receptors. As presynaptic DRD2 receptors exert an inhibitor control on dopamine release, increased availability of dopamine mediated by the T allele may contribute to the functional impairment of the dopamine reward circuitry observed in psychopaths, which may represent the neural substrate of impulsive and antisocial behavior of psychopathy. Concerning the TH gene variants, it is not known the functional effect. However, as this gene encodes for the dopamine tyrosine hydroxylase enzyme that catalyzes the rate-limiting step of dopamine synthesis, it may alter dopamine synthesis efficiency. In conclusion, we identified the genetic profile consisting of HTR1B GG genotype, ANKK1T allele, and THCG genotype that seems to make carriers significantly more sensitive to paternal maltreatment and that may be predictive of higher level of psychopathy. We believe that these data have important implications for both a clinical and a forensic perspective. Thank you.
Thank you very much, Dr. Palumbo, for this insightful presentation. As said before, we been holding the discussion at the end of the presentations. So now I ask you to stop sharing your video.
Sure. Yes.
And I leave the floor to Professor Heller. His presentation is titled Authenticity in the Ethical Debate on Memory Modification. Too early to forget about it. Please, Professor Heller, the virtual floor is yours.
Thank you, Andrea. It is great to be with you all, if only online. So let me get my show going. I will briefly be talking to you about the ethics of memory modification and more particularly about memory erasure. This is just a few ideas drawn from a book chapter that I'm working on. So first of all, to explain what I mean by memory modification, I refer to interventions that are aimed at directly altering people's memories. Usually, the basic purpose is to improve their well-being, although there could also be less palatable purposes. Typically, these interventions are biomedical in nature, although there are also other like behavioral types, for example. And the type of memory modification I'll be focusing on is memory erasure. So by this, I refer to interventions that completely remove a certain unpleasant memory from a person's mind in all the senses that memory researchers refer to memory. So for example, episodic memory, the memories we may have that are quite vivid with some sort of a visual imagery, for example, of a past experience, these would be erased. Semantic memories of the facts of that past event would also be erased, and also implicit memories, any implicit memories that would be present. So one example would be a fear memory. That would be a typical example of an implicit memory that could be sort of referring to a conditioned fear response to certain stimuli based on a bad experience that one had. All of this would be erased in memory erasure. And in the literature, in neuroethics on this, memory erasure is often contrasted with memory blunting. So memory blunting does not target episodic and semantic memories of past experiences. It's just meant to ideally remove the sting of bad memories, just make the person less upset when they recall the memory, especially in the case of traumatic or pathological memories. And it's supposed to do this by removing an associated implicit memory while leaving the episodic declarative ones intact. Then we also have, as other examples of memory modification, the implantation of false memories. That's really a staple of science fiction, as you will know, and memory enhancement as well, an important form that I will not discuss here. Now, in just a few words, why should we be interested in memory erasure from a neuroethics perspective? Why would anyone want to erase their memories completely as opposed to just blunt them if there is the choice between the two, right? Because erasure is a much more radical procedure. In a nutshell, erasure is more powerful, more effective than blunting, or can be expected to be such. For example, in the case of traumatic memories, it seems that it could eliminate the risk of relapse from a resurfacing of a suppressed traumatic memory, which could be an issue with at least some forms of memory blunting. Also, it has been proposed that you could erase the memories of violent criminals, remove the memories of their crimes. So I guess the idea is that you could reshape their self-image so that this would reduce the risk of recidivism. The person would no longer see themselves as a murderer in an extreme case, so they might be less likely to commit another murder later on. And otherwise, there's the sort of rationale that you may be familiar with from fiction, just a general idea of avoiding a pleasant affect caused by bad past memories, a bad breakup, as in the case of the movie Eternal Sunshine of the Spotless Mind, where the couple removes memories from their own relationship, their failed relationship. And perhaps you could create a more pleasant self-narrative by implanting false memories in conjunction with erasure. When it comes to the tools, scientific tools that are available to erase memories, they are still under research and development for now. But there have been some interesting results in mice. And I'll just mention one here, which is the latest kind of most cutting edge one, which is optogenetics, which is a technique that uses light signals, so lasers essentially, to activate and deactivate specific neurons. And there have been some interesting results accomplished in mice with this technique, such as the selective erasure of a memory, a reversible erasure. This was work by Steve Ramirez and colleagues at MIT. Also, the implantation of false memories was reported. And even though I am not aware of applications in humans of optogenetics for the purpose of erasing memories at present, the prospect has already been discussed, for instance, in a paper that was published in Age of Neuroscience earlier this year. So to move on to my main content here, so I want to look at this objection from authenticity, quote unquote, which is a sort of long-standing one in the literature. Already almost 20 years ago, it was discussed by the President's Council on Bioethics in their book Beyond Therapy, and I've discussed it, well, in a paper I published when I was still a grad student, and your own Professor Andrea Lavazza has also published on this topic. Now, what do we mean by authenticity? It's kind of an ambiguous term, but one relevant sense is this idea of truthfulness. So the idea is that, or the concern is that, if people start erasing their memories of bad experiences, their lives will be less truthful, they will be less in touch with their past, and that is a bad thing. That's the objection or the concern in a nutshell. And this concern has received some criticism. So one criticism I want to look at here is a skeptical response, which basically states that this concern is flawed because it overestimates the reliability of our autobiographical memories. So you have authors like Nada Gligorov who have defended such a skeptical objection. And typically they cite research like the work of Elizabeth Loftus, for example, which I'm sure several of you might be familiar with, which essentially shows the vulnerability of our memories to corruption or contamination. If we are subjective to suggestive questioning, or we interact with someone else who may have an inaccurate recollection of a certain event, our own memories might become less accurate, is what this body of work suggests. And from there, I guess you could develop this critique in two ways. A strong one, which I take inspiration here from the field of metaethics, so I call it the error theory version of the critique. So it basically says that this body of research shows that most of our autobiographical memories are inaccurate, in which case there is no truthfulness to be lost by erasing memories. In fact, we might even gain truthfulness by losing some false beliefs. That would be a very strong version. Then a sort of more nuanced version would be what I call a skeptical approach. So on this approach, one would say, well, this research shows that in any given case, we lack a justification, a good justification for trusting our autobiographical memory. Maybe it is in fact accurate, but we're not justified in believing that it is. And this is supposed to, kind of undermine this concern about truthfulness. So what should we think about this critique? So my reply is as follows. I think that this body of evidence, this important body of research, does show that our memories are fallible, that they are far from perfect. However, I do not believe that it justifies the error theory approach, the claim that autobiographical memories are typically inaccurate or always, or actually they're not always inaccurate. But I think it's actually, this claim is actually incompatible with the available evidence, partly because we know that there are differences in the ability to accurately recall, some past events based on the research that has been done, for instance, on flashbulb memories. Also, I think an important point here is that there might be some slippery use of language at play in this critique. So we have to be careful what we mean when we talk about an inaccurate memory, right? There's two things we could mean by this phrase. We could mean either a memory that's completely off the mark, that gets all the main details about our past wrong. Or it could mean that it's just not perfectly accurate, right? Maybe it gets the gist of what happened correct, but it has some details wrong. Maybe the color of someone's shirt is wrong or something like this. So that would mean also a memory that's inaccurate in a more moderate sense. But clearly we cannot, if we mean, if by inaccurate memory we mean less than perfectly accurate memory, we cannot say that erasing such a memory will not eliminate any sort of truthfulness or contact with the past that we may have, right? It could still provide some degree of contact with the past. So that's an important point to bear in mind here. Now, when it comes to the skeptical approach, which is, I think, more plausible than the error theory one, I still think that even if we assume that it is empirically justified, it's still not clear that it refutes the concern about truthfulness. Rather, at most, what it shows is that we might need to be skeptical about it. We might not know in any given case whether it applies or not. For all we know, maybe it applies, maybe there will be a loss of truthfulness, maybe not, because we don't know if the memory is accurate or not. But it's not the same as saying that the concern is invalid and unwarranted. But I would go further than that. I would say that, in fact, it's most likely in at least many cases in which people are likely to use this sort of procedure, assuming it becomes available and sufficiently safe to use. I think in most of these cases, we can expect that skepticism can be overcome because the events in question will probably not be purely private events. They will likely be publicly observed events that have some witnesses or maybe some kind of evidence, like a Zoom recording or something of this kind, that would explain why the memory is so charged, at the affective level. It's because, it wasn't just a private event. It was something that other people realized, and that might help explain why someone feels so bad about it. Now, to conclude, while I am not convinced by the skeptical objection to the truthfulness concern, I do believe that there are reasons to temper this concern nonetheless, which are as follows, I would say. The first one is that even if we do believe that this idea of truthfulness in memory is coherent and that it's something that's valuable in some way, at least instrumentally, to learn from the past, to learn from past experience, but also perhaps intrinsically because we think it's part of a good life in some way. Even if we do think that doesn't mean that it's a value that overrides all others, right? It could be that it is outweighed, at least in some cases, by other considerations, such as the pursuit of happiness or achievement, which could be facilitated by erasing some memories. I think even those of us who think that it's very difficult to outweigh this value will argue that they will agree that there will be some cases where it will be outweighed. And to add to that, I would say we also need to distinguish between two different kinds of memory erasure. The first one is what I call a soft erasure, where you just erase one episodic memory or a series of such memories, but you do not seek to suppress completely the awareness of the past act or experience. The person might still remember, might still have information about what happened, be aware of what happened, but just not vividly remember it from a first-person perspective. As opposed to what I call full-fledged or maybe hard erasure, where you try to completely remove all recollection of the event, so both episodic and semantic memories, and maybe you try to create a sort of alternative history for the person, which might actually require cooperation from other people. So perhaps an optimistic note on which to finish is that soft erasure poses less of a threat to the value of truthfulness than full-fledged erasure, and it also seems more technically feasible. That doesn't mean that full-fledged erasure is no concern at all, for instance, because it is required by some of the applications I've described. And we can imagine some authoritarian regimes being interested in it, but at least it is a more remote prospect, I think. Thank you.
Thank you very much, Professor Eller, for your presentation. As said, we can discuss at the end of all the presentation. And now I see Dr. Casartelli is a member of a research group, and he and his colleague Vitale, Federici, Pini, Molteni, Ronconi will be presenting a paper whose title is "To See or Not to See in the Same Way? This is the question. Inflexibility of visual scene parsing in autism." Please, Dr. Casartelli, the floor is yours.
Sorry for the audio. Thank you, Andrea. Can I share the screen? Yeah.
Yes, you can.
Yeah.
Yes.
That one. Condivide. Okay. I need to check my system preferences. Sorry. Sorry for this mistake. Usually we use Teams and Google Meet. So I have not the access to the other one. I need to left the talk 1 second. I return. Just a second. Sorry for this misunderstanding.
Okay. So I think while waiting for Dr. Casartelli, we can use this time, which is very precious, as you know, to collect some questions for the other speakers. Has anyone has questions? I'll be starting with a clarification question to Dr. Palumbo concerning the percentage of people who are in the class of those who have all the gene variants in your group. I mean, how many people, or which is the percentage of people who have the all variants?
All right. Yeah, that's. Yeah,
I think it's important because in that case the influence of paternal maltreatment is very high, and in the other cases instead is not so high.
It's not so high. That's true. So I didn't report the number in the presentation, and I don't remember the exact number.
But now you have the chance to do that because Dr. Casartelli is now back. So now he will be presenting.
In the meantime, maybe I have the chance to do that.
You can search for the number. Okay, thank you very much. And Dr. Casartelli, the floor is yours again.
Okay, do you see my slide?
Yes.
Okay, sorry again. First of all, please excuse me for the delay. Obviously, I'm not the guest star. I am just an Italian guy with too much confidence with the public Italian transport system. So this is the problem. Please excuse me for this delay. Thank you, Andrea, for this introduction. And today I would like to share with you the results of a recent study that has been accepted for publication at the end of the 2020 on Neuroimage Clinical. Last year in May 2020, this data was very fresh, and I was very curious to share these results in a non-completely neuroscientific scenario, such as the one that we can benefit from this Neuroethic Congress. But unfortunately, it was not possible. So today, these results are probably a little bit more robust in the sense that we have also further insights on this direction coming from other studies from our group. And I am happy to share with you this hypothesis, this line of research that I am pursuing at the Scientific Institute IRCS Medea. It is a pediatric research hospital specialized for neurological, neurodevelopmental, and neuropsychiatric condition. My research unit is named Theoretical and Cognitive Neuroscience Research Unit, and we are a small group working mostly on sensory, perceptual, and motor functioning in ASD, with a range of distinct techniques such as EEG, eye tracker, motion capture, and fMRI. The main peculiarity of our group is the theoretical counterpart in cognitive neuroscience, and notably its impact on developmental clinical neuroscience. A well-established view describe autism spectrum disorder, ASD, as a neurodevelopmental condition with heterogeneous phenotypical manifestation, impacting on the social domain and characterized by restrict and repetitive pattern of interest and activity. Beyond these aspects, and despite the growing body of studies investigating the pathophysiology of ASD, neurobiological underpinnings of the condition are far to be clarified. This is the classical framework that you can easily find in any developmental psychopathology textbook or in the DSM-5. In the last decades, the main focus of research activity in the context of ASD have been related to putative difficulties in the social domain. And this approach has been recently questioned, trying to address the bidirectionality of the social dynamics. For example, we have demonstrated with a combined motion capture plus psychophysical approach that the way in which you express your attitude with the motor system, for example, gentle, pass the pen in a gentle way or pass the pen in a rude way, impact the readability of your action. We referred to this concept with the notion of motor prosody or vitality form. We found that children with ASD and neurotypical control express, for a motor perspective, their attitude in a different way. That is, the modulation of distinct type of motor style is predicted by different kinematic parameters in the two groups. And this is reflected by difficulties that neurotypical adults show in understanding action expressed by children with ASD compared to action expressed by neurotypical children. This means that it is not enough to study putative difficulties of children with ASD in understanding neurotypical actors, but we need to focus also on potential difficulties that neurotypical individuals have in understanding individuals with ASD. More simply, we need to take into account the bidirectionality of social dynamics. This new line of research contributes to support the idea that social interaction is a complex and multi-layer phenomena that should be taken into account for multiple perspectives. In line with this idea, today my focus is on basic perceptual function, and notably on potential cascade effect of anomalies in this basic perceptual function or more complex ability. Recently, a specific focus on sensory and perceptual processing anomalies in ASD has been proposed in the literature. Clinical and research perspective agree in stressing basic sensory and perceptual mechanisms as core components that characterize early neurodevelopmental derailments in ASD, with potential cascade effects on the development of more complex functions. In parallel, recent evidence in neurotypical population ascertained the critical role of temporal dynamics in driving sensory and perceptual sampling, notably modulating the visual scene parsing that, in turn, impact on our visual understanding of the world. Noteworthy, neurotypical sensory and perceptual parsing seem to be characterized by flexibility and modulability. Here, we benefit from a visual crowding task to test flexibility versus inflexibility of neural and oscillatory dynamics in visual scene parsing in typical developing and ASD children, combining well-controlled psychophysical procedure and cutting-edge cognitive neuroscience analysis. Visual crowding is a perceptual phenomenon consisting in a decreased ability to discriminate objects when presented with distractors. Crowding limits the recognition, but not the detection, of visual stimuli of different complexity, ranging from simple objects such as orienting graphics to more complex objects such as letters or faces. More simply, visual crowding may be considered an efficient way to test the dynamics between visual information processing and distractor suppression. Anomalies in this dynamic may have relevant cascade effects in the efficiency of the visual scene parsing. So the visual crowding task was proposed to a group of children with ASD, and an age and functioning matched group of TD children, TD peers, was also recruited. In the table, you can find the clinical future of participants. Participants had to discriminate the target letter surrounded by flankers at different distances. In the strong crowding condition, Flankers are near to the target and the recognition task is supposed to be more difficult. In the mid-crowded condition, between target and flanker, there is a filler that should simplify the recognition task. Finally, we set also a no flanker condition as control condition that has been considered as the baseline for analysis. Simultaneously, an high density electroencephalography registration was provided with 64 channels geodesic system. Behavioral, that is accuracy basically, event related potential ERP and cortical source reconstruction analysis were performed. At the behavioral level, both ASD and TD group show a significant influence of the level of crowding on the accuracy rate. This means that in both groups, as expected, stronger crowding leads to lower accuracy in the recognition task. This pattern of accuracy is consistent in both groups and directly confirming that children with ASD perform appropriately the task and understood the experimental request. A different scenario is reported for electrophysiological results. At the ERP level, occipitoparietal sensor revealed a significant modulation of the N1 component according to crowding regime in the TD group. In contrast, this modulation was absent in the ASD group. Being N1 a component that previous studies associated to visual crowding tasks in neurotypical adult population, this result suggests that neurotypical children at the ERP level show a pattern consistent with neurotypical adult population. At the cortical source level, the activation of occipital, superior parietal, and inferotemporal regions was modulated by crowding regime in the TD group. In contrast, this modulation was absent in the ASD group. To decompose complex constructs such as social cognition into more basic components such as visual processing, integration versus segregation processing, reflects the need of taking into account the neural and oscillatory counterparts of specific function. This is even more urgent when we deal with clinical and pediatric population in which the developmental trajectory are, by definition, atypical. Neurotypical brain does not sample sensory and perceptual information in a rigid way, as you can clearly see from the ERP and source reconstruction analysis. Neurotypical brain efficiency reflects the ability of using stable rule under fixed condition and the ability of changing them rapidly according to the contingent physical and contextual future, such as, for example, distal level of crowding. Behind approaches described ASD as a constellation of heterogeneous behavioral symptoms, nowadays multiple lines of evidence converge in suggesting inflexibility and rigidity as critical feature of ASD. Although many differences may characterize each approach, a general promising idea is that inflexibility is not circumscribed into a specific domain, whereas it may spread in a different sensory and perceptual sphere. Our present findings nicely fit with this idea. Despite a comparable behavioral performance between the two groups, our study demonstrates that children with ASD and TD controls fulfill the task with different oscillatory and neural costs. Even more intriguing, as compared to the control group, children with ASD seem to be driven by a different sampling strategy. In line with previous findings in adult population, in the TD group, the mean amplitude of the N1 component was significantly suppressed in the crowding regime. Furthermore, in agreement with the general idea that neurotypical brain efficiency is mirrored by the flexible and modulable recruitment of neural resources, in the TD group, the cortical reconstruction show that the activity of the occipital, superior parietal, and inferotemporal regions was modulated by the level of crowding, as you can see in the maps. The scenario was different for the ASD group. Children with ASD recruited oscillatory and one component and neural resources, occipital, superior parietal, and inferotemporal regions, in a comparable way, regardless of the crowding regime. Such results may indicate that in TD children, the neural cost is well balanced according to the task demand, Whereas in children with ASD, viscosity is rigid, inflexible, and finally not balanced conveniently among different crowding regimes. Accordingly, we are tempted to suggest that the controls follow a balanced sampling rule, alternating stability under fixed condition and flexibility when something changes. Whereas children with ASD follow, at least in this task, a rigid, inflexible, and non-modulable sampling routine. This represents a promising candidate hypothesis to explain anomalies of visual scene parsing in ASD. To tackle sensory and perceptual anomaly in ASD for multiple levels of analysis represent a fundamental step forward to deeply characterize the visual experience and the visual understanding of the world in ASD. Our results suggest that neurotypical participants recruit neurooscillatory resources according to the specific requirements of the visual scene. In contrast, children with ASD seem to be characterized by a rigid, inflexible, and non-modulable neural and oscillatory costs. These results may also spark new insights to understand the core phenotypical manifestation in ASD. Indeed, an inflexible parsing of the visual scene may lead to a different comprehension and in turn interpretation of the visual environment. Following this hypothesis, atypical behavioral responses that are often considered the core feature of ASD may simply be coherent with their specific interpretation of the visual scene. Here, I want to be clear. I do not intend to claim that anomalies in visual processing can explain all clinical manifestations of ASD. Neither that visual processing anomalies have a sort of prominence as compared to other candidate markers of ASD. Nevertheless, state-of-the-art literature on visual scene parsing in ASD and our own studies strongly suggests that the heterogeneous clinical manifestation of ASD may be explained, at least partially, by anomalies in processing and encoding basic features of the visual scene. This represents an intriguing paradigm shift both for clinical and the research perspective, and it is in line with the idea that you do not only see a picture with your eyes, but there is a world behind your visual perception of this scene. And this is totally consistent with an Husserlian phenomenological view of perception processing. I would like to thank Andrea Vitale, postdoc in our unit, Monica Nicoli, research assistant in our unit, Lucia Fumagalli, clinical psychologist, and Laura Villa, neuropsychiatrist, that are the real core of our group, and their contribution is obviously crucial. And thank you for your attention to it. I hope you have enjoyed this presentation.
Thank you very much, Dr. Casartelli, for your talk. Very, very interesting. And now we have some minutes for questions and discussion. We have a question open with Dr. Palumbo, if she can now answer.
Yeah. So I found the number of subjects that have the complete genetic profile that increases the risk of psychopathy, and they are 25 subjects.
25,
so 25
out of how many?
Out of 232.
I see.
Yeah, so these are the subjects with higher risk for psychopathy, and among these 25, half of them have low risk of psychopathy because they have they were not maltreated by the father, and the other half have high maltreatment scores, so they also have very high risk of psychopathy.
Thank you very much. Now we can get questions for both Professor Heller and Dr. Casartelli. I have a question to Professor Heller. When it comes to erasing memories, I think, and important feature to consider is that many actions or deeds by us are, especially today, documented or recorded in some ways. So how can you deal with this problem when you try to erase a memory?
Thank you. And I agree that this is potentially a challenge for the what I've called the full-fledged memory erasure, that particular goal. I think, of course, there are ways also to erase data, so some people try to do that, try to erase historical records. So lots of things can be erased, but if you have protections for historical records and things of this nature, and of course if you have eyewitnesses, yes, it complicates this task of removing some particular event completely from someone's memory. I think one of the authors who was considering this prospect was saying you could perhaps, like when it comes to a criminal, for example, you could try to send them to a different setting. Like the analogy was with eyewitness protection programs. So I suppose the idea was to give them some entirely new identity and put them in a different setting where they would not come into contact with evidence of what they had done. But of course, that seems quite sort of a fragile, endeavor. It would be very difficult to guarantee that the person would not come into contact with this evidence. So to some extent, I take this to be a reason to mitigate the concern, because it's not so easy to rewrite history, rewrite one's personal history, especially in liberal democracies. Yes.
Yeah. In some way, allowing memory erasure would entail to erase public data, a duty to intervene in society in order to make the reality consistent with the personal memory of the person.
Yes, I mean, maybe to just raise one point to make this prospect sound maybe less absurd than it might otherwise. So we do have some debate, for example, about things like the right to be forgotten when it comes to search results on the internet, and there is some sort of forces pushing in two different directions. You do have people who want some information not available to the public because they think that it hampers their own life prospects and it's not in the public interest. But then where do we draw the line? For instance, when it comes to the public's right to know, I think some search engines have come under fire, I believe, in the past, have been criticized for being too quick to comply with some such requests. So there are disagreements to some extent about how much information we should keep, protect, to what extent people should be able to remember certain things. Of course, this is not as extreme as in the type of cases I've described, but yeah, I guess sometimes I am surprised by the way social mores evolve. So I guess nothing can be completely ruled out. But I agree with you that would be a big challenge there.
Thank you. And to prove Dr. Casartelli, I'd like to know if you see some ethical issues or implications with your research.
For sure, it is not easy to link basic research such as this one with a direct ethical implication. The risk is that we can misunderstand the link, and I'm not sure that the best way to characterize this research is to find a direct link with an ethical problem. In contrast, I think that it is very, very urgent to use it to do strong and well-organized, well-balanced, well-controlled basic research to better understand the future ethical implication in clinical psychopathology. Because the problem is not to find the link for any study to an ethical problem, in my opinion. In my opinion, the most important thing is that we need to understand the condition. For example, the point on the bidirectionality of the social dynamics, for sure, it has a potential implication for the understanding of the behavioral manifestation. And so the point is interesting if you, in my opinion, if you consider the fact that you have to understand the condition before setting the ethical problem.
The ethical issue I see is that, as you very well showed earlier, is that we have many strands of research on ASD, and when it comes to make a decision for health policies, for example, we need to choose between different interventions because we have a limited amount of money, for example. So people who have the responsibility of making this choice should have a very good knowledge of all these strands of research in order to choose the best one. It's not easy at all, I think. So there is an ethical conundrum in choosing the best intervention based on the best research. And when we have many very recent findings, it's very difficult to make this choice, and I think it's also an ethical issue. Do you agree?
Absolutely, absolutely. Another point that may be of interest for us in this Congress, I think that we need to accept that there is not ASD today. This view of ASD will not be the same in the future. Probably we will know better the pathophysiology of the condition, the clinical manifestation, the comorbidity problem, the age-dependent clinical manifestation. So we need to accept the fact that you have to challenge the research activity on ASD, and policymakers have the need to see the variability and the new challenge of research line. This is true for ASD, but I think it is true for every clinical condition.
Thank you very much. Any other questions? I cannot do all the questions myself, so please, if there are any other comments or remarks on these three very interesting presentations. Otherwise, Alexander, please.
Thanks. Can I ask a quick question to Sarah? Just regarding your final point about the implications of these risk factors that you have uncovered. I was wondering what would you describe as the main ones, the main practical implications? So for instance, would it be in terms of genetic counseling, or in that context that some advice could be given to try and avoid bad outcomes if parents are better informed, or would it be some other context?
I think that probably the main implications will be on two levels. One, maybe an implication that could help in selecting those subjects that are at higher risk in jail, and maybe try to shape a specific treatment to in trying to help these criminals with this particular genetic profile. And on the other side, the implication would be before they are they're put in jails. So during the decision and so for, as you were saying, in genetic counseling, in trying to understand how much the DNA, his DNA and his experience had a role in what happened.
Thank you.
Thank you.
So I think we need to conclude our session. Don't miss the next event at 8 p.m. just time. You should have received the program with the relative link. This is an event organized by INS on psychiatry in the era of COVID-19, the so-called telepsychiatry. It's a very, very interesting event. And tomorrow at 5 p.m. we'll have Vittorio Gallese and the Cine Assembly at the end. Thank you for your participation. Obviously, thank you very much to the presenters, and see you soon.
Thank you so much. Very nice seeing you again.
Thank you. Bye-bye.
Thank you.
Bye-bye.
Bye.
Bye.
Bye.